we're going to talk about three main approaches to identification of microorganisms, and by no means those approaches, what I'm telling, is comprehensive and covers all possible methods. I just handpicked 6, either because they are like very foundational or they're really exciting of sorts. And the first big approach that we're going to be talking about is immunological approach to microbial identification. So all immunological methods are based on the fundamental principle of the recognition of the antigen by the antibody. So imagine that you have a molecule, usually it's a protein, okay, that is foreign, and that is important. What is it foreign for? Whatever. For instance, let's model all this conversation using the elephant in the room, coronavirus, and the spike protein of coronavirus, okay? Spike protein is in the envelope of coronavirus, and if you will stick that spike protein into, let's say, sheep will produce antibodies to it. So that's our spike protein. These are sheep antibodies recognizing it. Okay, we're good? The very important feature of the antibodies is that they are highly specific. And we need to remember that all the time. So now imagine you are at the very beginning of the pandemic, so you don't really have any diagnostic tests yet, and you have a patient who succumbed to pneumonia. So you want to know whether or not this patient had this particular, you know that can be coronavirus, and you do have antibodies, those specific blue antibodies to coronavirus spike protein. Are you following me? Now you want to know if the antigen, the spike protein, is in the lungs of that patient. Remember, patient's dead, so you can take a fragment of the lungs. So take a fragment of the lungs and you put it in the tube, okay? So put it in the tube and you make Lysa, like you homogenize it, spin all the trash down, so now at the end of the day you have liquid, okay? The main question is, how do we recognize that this liquid contains this spike protein? Is it there? Because this liquid will contain a whole bunch of other proteins, right? Like it's just a mixture. How you parse out just one? One really good way is to separate them. And how can you sort them out? You can separate them based on the size, okay? So far, does that make sense? Now, you know the size of that protein, right? So imagine you have a crowd of kids, and you know that the kid you're looking for is, I don't know, 80 pounds. In that crowd, you have kids with a body weight from 40 pounds to 150. Are you following me? They're all crowded together. You can't really pick your kid, okay? But can you separate those kids based on their body size? You can. What you do, in my example, proteins are kids, you prepare what's called a gel, and it is literally a gel, okay? And you load your sample on the gel. You apply electrical charge to the gel, and proteins that are negatively charged start moving down here. That makes sense? Now going back to kids. If you have a whole bunch of kids, some of them are really slim, like 40 pounders. Some of them are really, really big, like 150 pounds. How do you separate them without having scales? You force them to run through the really dense forest. Small kids will sneak between the trees, right? And the big fat kids will get stuck between the trees. You see, the smaller will move faster. Same goes with proteins. Smaller proteins will move faster. And you will be able to separate your sample, proteins from your sample, based on the size. Okay, we good? Presumably, your protein, this one, is somewhere there, like here. Technically, at this point, you may say, okay, let's just slap antibodies onto this gel and just look at it, right? It's not going to work because gel is insanely inconvenient to work with. So what we do, we take the gel and transfer the content of the gel onto the membrane. How we do that? We take the gel, put the membrane on it, and apply electricity. And this separated pattern of proteins just transfers like a Xerox copy on the membrane. This is why this method, is called Western blotting. If you want, I can tell you why it's Western. If you don't really care, we'll skip. It's a stupid story, has nothing to do with the technique. Okay? Blotting is important. Does that make sense? So what you end up with is a membrane which looks like a piece of kind of delicate paper with all the proteins separated on it, including your protein of interest. Now, can you see them? No, you can't, because the quantities are really small. But what you can do, you can take your antibody, the solution of antibodies, apply it to the membrane. So these antibodies now bound to that protein. And then it becomes really a technicality, because all you do, just, you have some kind of a signal at the end of the antibody. When I say signal, what do I mean? When you apply this antibody, if your protein is present, there will be a color change, like a little band. Or there will be a luminescence or fluorescence. Does that make sense? Some kind of a signal. Doesn't matter which one. As long as you can detect it, you're fine. Am I clear on this one?
I understand that whole nasal swab card now.
We'll get there. We'll get to the nasal swab. We actually will get to the two tests that are used in the context of coronavirus infection. That I can tell. Okay, so this is why I told you, we in the beginning of the pandemic, we don't really have any commercially available tests. So why I used Western blot? First of all, fairly easy to do, fairly easy to learn, very adaptable to new antigens and new antibodies. There are a few disadvantages though. Technical disadvantage, it's long. Probably would be a bragging to say for an experienced Western blotter like me. I did a lot of them years ago, like 10 years ago, I did a lot of them. But from the moment you get the sample to the moment you get the result, it's 10, 11 hours, easily. You make everything from scratch. It's just tedious. It's fun, but it's tedious. It's really cool benchwork. Second problem, you can find only the things you're deliberately looking for. If you don't find spike protein, you still don't know what your patient died from. You see what I'm saying? Like, you don't get an answer. If you found it, that's cool, but if you didn't, that's a problem. Does that make sense? Now, can we improve something about it? Well, the answer is yes. We can do what's called Lysa. It is not, it is female name, but in this context, the Lysa is an acronym. Okay. Again, I'm not going to ask what it stands for, and I believe a lot of people who use it, they may have forgotten what it stands for, but everybody understands what it is. So, this is something that can be used for testing for the antigen, but it must be used at really, really large scale facility. You're not going to use it. So what you refer to as a nasal swab, that's a different.
Well, it's like rapid, like a rapid
Oh,
COVID.
this is not rapid. If you want, I can very briefly explain how rapid antigen test works. That I can do. Okay? Let's do a Lysa, and then I will put the rapid antigen test next to it. So you have a plate with wells. I just put 3 wells because it's going to be easier to exemplify. Make sense? So at the bottom of each well, you have antibodies. Okay? You have antibodies. And you add your samples. So I deliberately have 3 samples. One sample has 2 antigens. That make sense? Another sample has only 1 antigen, and the third sample has no antigen at all. So this fellow is fine. Okay, we're good? What you need to do next, you need to add another antibody with the signal on the back. So you're going to have so the signals, okay. Here you're going to have slightly less signal because you have only 1 antigen, right? And in this well, you're not going to have a signal at all. Are we good so far? So the more antigen you have in the sample, the stronger the signal is. Did we solve the problem with brain fart, I'm sorry. Did we solve the problem with you finding only what you're looking for? No. We still, if it's something else, maybe this fella is really, really sick. This fella just doesn't have that antigen, the red antigen. Maybe, it's something else, flu. We do solve a couple of problems with the Lysa, though. Solve the problem of the time, and we solve the problem of processivity. It's much faster. This can be automated, and you can do, like, the standard lab test is 96 wells on one plate. There are 384, and there's some pretty fancy plate that has 1,500 samples on it. Like, insane, okay? So if you do like 1,500, then it's automated, obviously, but you can do a shit ton of samples at the same time. Does that make sense so far? Now, I'm sorry, I will get my name cheat sheet, okay? So what Catherine was referring to is a rapid antigen test, which is based on exactly the same principle, but they have antibodies that immediately produce signal. You do not do any washing or anything. The good thing about that test that you refer to is that it's fast. The bad thing, well, kind of bad, it's really not quantitative. It just tells you yes, no, okay? So the idea is that if you have an antigen in your nasal sample, it will interact with the antibody, usually on some kind of a carrier, like a membrane or a paper, okay? And this antibody, as I mentioned, will have a signal, okay? And only after the interaction, the signal will kind of show up, okay? That's it. If there is no antigen, there is no signal. There is some kind of a buffer that you put on it, and that buffer only in the presence of the complex will kind of trigger the signal. So fast, yes. sensitive, yet, well, reasonably so, and quantitative, no. That's the rapid antigen test. So far, you following me? Now, one more test that I wanted to go over here is immunofluorescence, which is really fun. So what is immunofluorescence? Sometimes you are not so much interested in finding how much antigen you have, but you may be more interested in finding where is it. So again, you have your patient. So let's say you have your patient, you detected the antigen in the lungs, okay? Then you decide, okay, I'm going to develop a Lysa. You developed a Lysa, now you can quantify how much antigen is in the lungs. Are you following? Now, last bit of information that you want to take out of that patient is where in the lungs this antigen is. So you take a sample, take a biopsy again, okay? Well, it's not biopsy anymore because the person is dead. So take a lung sample and you prepare a microscopic slide with a slice of the tissue, right? And what you ultimately see on that slide is a whole bunch of cells. And if the antigen is inside of a particular cell, or between the cells, in the tissue, once you add antibodies here, they will bind to the antigen and they will produce a signal. Does that make sense to you? So, what is the common feature of all three methods? They're all built on the antigen-antibody interaction. What is the difference? Here you detect your protein after the separation on the gel. Here you detect your protein in the well by washing off all the crap. Here you detect the protein in the cell using fluorescent antibodies or in the tissue. So this method will not give you a quantitative response, but it will give you the answer to where in the lungs. For instance, you look at it and say, oh, that's, type 2 alveolar cells are infected or type 1 alveolar cells are infected. Okay? Questions on this one? Go ahead.
They're dead or not dead?
Huh?
They're dead?
Cells?
Not that person for the immunofluorescent.
This one?
Yeah.
Don't have to be.
Okay.
I mean, you can, for instance, if you have a patient, like you have a COVID patient, and you want to know if that patient has, I don't know, coronavirus. I'm trying to think about a tissue that you can fairly easily biopsy. Muscle. You can biopsy muscle, okay? Take a sample of the muscle from the living patient, and you slap it on the slide and you look at it. That's easy to do. Make sense? Like, you can see where it is. So far so good? Okay, moving on.